From Microbe TV, this is Immune, episode number 29, recorded on March 31st, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello, everyone. I hope everyone's healthy. I'm excited for this one.
We haven't talked in a long time, and such has changed since then.
It has. Yeah, a lot has changed.
Also joining us from Durham, North Carolina, Steph Langle.
Hey there. Glad to be here. Also healthy. But yeah, it's been a while. I think our last one we recorded the beginning of February, like the first week, and now we're at the very end of March.
Wow.
That's too long.
That's too long.
It's like a different era.
Well, yeah, considering circumstances.
Joining us today on Immune from Madison, New Jersey, Brianne Barker.
Hi, everybody. It's great to join you guys and to be here. I hope everybody's staying safe, and I'm excited to talk about immune responses with you all.
Well, we are excited to have you. This is awesome.
Yeah, I'm loving this crossover event. That's what I'm calling it.
Yeah, I'm doing crossovers on the pods, and I think it's good. And so thanks for coming, Brianne. And,
no problem. I'm excited to be here.
I'll just step back and let you guys all talk if you like.
Well, we had no plan for this. So.
Yeah.
Okay.
Brianne, you could introduce yourself to our listeners. I'm sure we also have crossover listeners, but your background and what you studied and what you do now.
Sure. So I'm currently a professor at Drew University in Madison, New Jersey, which is a small undergraduate college where I teach undergraduates and do research on innate immune responses to viral infection. I trained in HIV immunology and vaccines, getting a PhD in immunology in the past, and am a Duke alum. So I'm very jealous of Stephanie getting to be in Durham.
A bit warmer. And you were in the institute I'm in, DHV, the Human Vaccine Institute, correct?
I was actually an undergrad there before it became the Human Vaccine Institute. Or around the time that the name changed. And then I was a graduate student in a lab at Harvard that collaborated with you all. So
oh yes, nice.
And how did you end up on TWIV, Brianne?
That's a good question. I met Vincent at a conference and we just started talking. He invited me to be on an episode of TWIV once when I came to visit New York. And then I started subbing in for other hosts when they were sort of not around. And then I just sort of became part of TWIV.
Yeah. A good
You're
part.
kind of like a black hole. You can absorb everybody around you, right?
But it's nice to have you on there. I mean, one, you have a great teaching background being at Drew University, so it's really helpful. Not that the podcast didn't do that, but it's a nice voice. And also it helps balance out gender, giving it more of a, another female perspective in turn with Kathy. So I love having you on.
Thank you. It's sort of funny because in graduate school, I was the immunology graduate student in a lab that was all virologists. And so I've always felt like I was sort of on the borderline between being a virologist and an immunologist. So that's also, interesting that I ended up with TWIV.
And it's funny, but that's how we met, so it's fine.
Yeah, that's great.
So two things. First, Brianne's sister does the transcripts for Immune.
Yes, that's right. Thank you to her.
Cool.
It's very nice.
And
Absolutely. Thank you.
I got one last week and I haven't posted it yet. I'll get to it. Sorry about that. And then we have a lot of new listeners over on TWIV, of course, and someone wrote that they started listening to all the other podcasts, Immune, TWIP, etc., TWIM. And they say, how do you get all really good people who are clear and explain things really well? And I said, I'm really lucky.
Oh, well, that's a nice compliment. That's what we strive for.
Yeah. You screen heavily.
you get people who want to do it to begin with.
Yeah.
Because not everyone wants
I think that it's a really good group. I mean, in terms of just the dynamic of people, and we'll see where it goes. We got to get you more help though. You are doing quite a bit. You're pumping out podcasts like no one's business.
Yeah, so for those of you who are checking in this one because it's on the topic of the day, you should go over and check out Twit because Vincent's been churning them out like crazy, and it's such amazingly good information.
Yeah, everyone really appreciates how much you're doing, Vincent.
Well, thank you. Yeah. I appreciate it. I'm getting cranky lately, so if I sound cranky,
somebody tweeted. They said, "Tell Vincent he's allowed to take a break," and I was just like, "No. This is our glory day. This is when you know something and you can bring people together who can speak intellectually about stuff in a way that people can understand. Still, it's so very important. I mean, the science communication is more important today than almost ever before."
For sure. It's funny how now professors are starting to get this. Like last week on Twiv, we heard of an effort at Arizona to do Zoom sessions every Friday for the public, and there's an immunology one apparently, and I think it's great. It's like, we've been doing this for 12 years and been, I would say, in the scheme of the world, relatively ignored, right? Because we have a lot of great listeners and stuff, but, you put on a Netflix and millions of people watch it, but we can't even get close to it. But now we're sort of getting better because of the moment. And
yes, and what a moment it is.
I do think that scientists should realize that you always have an obligation to talk to the public, not just in the times of a crisis.
Yeah.
You got to write an opinion article, send it to Science, and tell them, this is great. We're all communicating more, and here's where it all started.
Yeah.
Well, I complained to my own medical center. I said, look, you send out these daily updates. You never mention that you happen to have a faculty member with all these great podcasts. Yeah, so finally they're doing something. But my gosh, you have to pull teeth. It's crazy.
It's crazy.
Anyway.
Yeah, and it's a shame. I keep noticing as I talk more and more to different groups of people, how many misconceptions there are out there. And I'm sort of thinking wow, we really need to do a better job with this. And I'm realizing again and sort of recommitting my efforts to science communication as I realize how many things people are not necessarily aware of, but also hungry to learn about.
For sure.
Yeah, we end up, in our own little world, and we're reading things on Twitter and Facebook and wherever else, and we think, gosh, how do people not know this? How do they not get the message, right? But clearly, we just needed to go in as many different media and as many different formats as possible, because you just never know what's going to catch a person.
Right. And what they're getting bombarded with are mixed messages. I mean, if their typical daily routine does not involve podcasts and involves TV, I mean, they're getting a lot of bad info. And so that's where we really have to try to break into their routine of listening to us. Yep, for sure. Well, we can start off by talking about, kind of the elephant in the room, and what we're going to really focus on today is the current pandemic, the SARS-CoV-2, or severe acute respiratory syndrome, coronavirus 2, that emerged out of Wuhan, China. I think the WHO posted in December 31st of 2019, there were some U.S. scientists that had been in communication that knew about it beforehand, likely was circulating for a while in December. And we as a country, maybe we're not as quick to react and be prepared. And so now what we're observing in the United States is spread in transmission of the virus. And then what we are doing now is basically lockdown and social distancing. And really, the economy is going to have a hard time with this. And I think that it really is a testament to investment in science. And I'm sure we'll circle back on that. But we can talk about how all of our lives have been impacted, but then we'll really hit into the immunology of this disease and some of the mechanisms.
Okay. I just want to point out, by the way, it's amazing how the clinical and basic science individuals in this country in the world have stepped up to the problem. You see stuff happening on an hourly basis. Clinicians are not only taking care of patients, they are developing therapeutics. They are testing them. They are figuring out indicators of severe disease, for example. And then the basic scientists are coming up with new therapeutics and new ways to assay. It is remarkable that so many people have turned to this virus and disease, right? I think this is the untold story, and it needs to be told, because we have a really good science, clinical science and basic research science establishment in this country. Could be better funded, but it's pretty robust. And look at how it's turning to this. It's really, to me, that is the amazing part of it all.
Yeah, I agree. I mean, I know my boss is an MD PhD, Daniel Griffin, who's on Twit, MD PhD. So it's been really refreshing. And I think we need to, collaboration is going to really what is required to do something about this.
By the way, immunologist, Daniel just sent me a graph of 100 patients that he's studied, where he's measuring the neutrophil lymphocyte ratio.
Mm-hmm. Mm-hmm.
And he said this is a predictor of serious disease.
Yep.
So he has 100 patients, 6 of them, a low, I don't know what the numbers, the ratio. Okay, here's the ratio. When the ratio is 6, the patients do well. When the ratio is 10 or above, they either go to the ICU or they die.
Yeah.
Why is that? Why would that ratio be a predictor?
So the neutrophils cause a lot of damage. And when you have lymphopenia, but too many neutrophils, they're going to cause more immunopathology. I don't know what the other people think on this, Steph and Brianne. What do you think?
I completely agree. I think that it is sort of some additional pathology coming from the neutrophils, maybe a little bit of a reduction in regulation coming from some of the other types of cells like the T cells, but it's mostly immunopathology, I would imagine. And I actually read a couple of papers preparing for this and saw similar kinds of data in a couple of papers, but none of them had as large of a number of patients as Daniel is reporting here. So that's really great data. That's really helpful.
Yeah, and it really speaks to, I mean, we can jump into the immunopathology of this particular virus and maybe other coronaviruses if we want. Or we could start with just speaking to the virus itself. I don't know how you guys want to work our way through this.
Well, I think, we've got 3 basically immunovirologists or virologists on here, and I'm not. But why don't we just start by talking about what kind of virus this is exactly and how it gets in, and then we can sort of go into how does your immune system know it's there and what does it do when it sees it?
Sure.
Yeah, sure. I think, Steph, you made this wonderful outline for us, so let's follow it.
Yeah, I think that's great. So this coronavirus is a single-stranded RNA virus. There are other RNA viruses like influenza and respiratory syncytial virus that also cause respiratory issues. But this is a really interesting virus in that it's found in many different animals. You'll hear a lot in the media and in scientific communication that this likely came from bats. And that's not something unique to this coronavirus. This is something that we see with the 2 previous human outbreaks. So that would be Middle Eastern Respiratory Syndrome coronavirus, or MERS, and then SARS before that in 2002. And there were a couple reasons why those were not as big of a deal as what we're dealing with now. Mostly because the pathology was so extreme. You could get people in the hospital. You can control it within, and not getting this community spread. But those also came from animals. And this virus potentially jumped from bats, maybe not. It needed even an intermediary species. It was ready to go. And it is able to bind to a particular receptor in I just thought it would be lungs, but it looks like even our upper airways and our nasal mucosa. And that's called ACE2. And that it's an enzyme, angiotensin converting enzyme 2. We could get into angiotensin, the pathway if you want, but that's the receptor it gets a hold of to be able to enter cells.
Yeah, and that, as Steph mentioned, that receptor, ACE2, is in a lot more types of cells than receptors for some other similar viruses. And so usually viruses like this maybe have a receptor in the upper airway or the lower airway, but this receptor seems to be sort of everywhere throughout the lung, and in other parts of your body. It may even be in aspects of your GI tract, for example.
Right. Which makes it a bit problematic if we're talking about the spread of this virus, because we're seeing a lot of reports potentially in stool. That was something that was also seen with SARS. But the fact that it's happening across age groups in terms of stool shedding can make it problematic.
Yeah, we have to learn still what fraction of the infections are transmitted by fecal-oral contamination. I think that's important. But either way, good hand hygiene will help, right, from both respiratory and fecal transmission.
Yeah.
Yeah. And what's interesting about this virus is it needs kind of two steps to be able to enter your cells. So we talked about ACE2 as a receptor. All viruses have receptors, but it also needs some type of host protease to be able to cleave a very important protein that we'll mention throughout the podcast called the spike protein. And that spike gives it its characteristic shape, looking like a crown, why it's called corona, and is really the driver for neutralizing antibodies, something we'll mention. But the host protease can cleave that, allowing membrane fusion and further entry into endosomal bodies inside cells. All targets
so that's
for
important,
drugs.
right? Because there are two types of viruses. There's enveloped and non-enveloped, and that has to do at least. Whether it's enveloped or non-enveloped determines how stable it is too, because we talk a lot about that. There's a lot of talk about that in the media, that these viruses stay on surfaces for a really long time. But in fact, enveloped viruses are less stable. Isn't that right?
Well, they tend to be in general, right? The coronas are unusual, as Steph well knows, because there are many that we know of that can pass into the gastrointestinal tract. And if somehow this enveloped virus withstands the stomach, low pH and enzymes and all of that and still infect the GI tract. So that's why we're seeing it there. And it goes against conventional wisdom that envelope viruses don't infect the GI tract, but they clearly can depending on how that is constructed, I guess.
Yeah, I think that this virus, it's the largest RNA virus within the RNA virus family, and its replication machinery, which we may not touch on, but it's pretty special in the way that it's able to accommodate mutations and pick up pieces from other animal coronaviruses or human coronaviruses. And so that it just has a lot of tools in its toolbox to make it potentially, like you mentioned, Vincent, the virus immunology I worked on my PhD was a gastrointestinal, an enteric coronavirus, and it did just fine. It did very well at transmission.
In fact,
so I don't want to take things. Oh, go ahead. These
viruses originated in bats, and in bats, these viruses cause GI infections. So they're really good at that. Yeah.
So I was going to ask sort of a little bit out of order. You mentioned about they're using this special machinery and they can have mutations. That's one of the things that people have a lot of fears about, right? We know a lot about flu and how it changes from year to year. Can one of you guys comment on how much we know about how this virus changes, and could we expect major changes over time?
I have some thoughts, but Vincent, as our expert virologist, I can let you start.
You have actually worked on coronaviruses, Steph, so you're the expert. But I think that there's not the kind of antigenic variation we see with influenza virus. The coronaviruses seem to be relatively stable antigenically, and so if we are able to make a vaccine or vaccines at some point, I think they will be effective for many years. What do you think, Steph?
Yeah, no, I agree. And I think one of the interesting things that coronaviruses have evolved to have is something called an exonuclease. So most RNA viruses have a mutation rate that they're unable to deal with. And so it results in a lot of different mutations that mostly are terminal. I mean, mostly they're not going to wind up in a productive viral infection, but sometimes they do. But with coronaviruses, they have the ability to correct themselves. And they also have a very different polymerase because of that exonuclease. So those are two main things. We think that the exonuclease has allowed then this genome to just become very, very large. And you'll see, and I don't think we even fully understand how these viruses persist amongst animals, but they can last a long time in cell culture without accumulating large detrimental mutations. And it kind of speaks to their ability to transfer between animals for a long time until they eventually get a mutation that potentially would change the protein enough to bind to a slightly similar receptor, but in a different animal. So
I think that this idea of an error correcting machine is interesting because as Mark Dennison told me last week, and that was a TWIV episode, it makes making antivirals that target the polymerase difficult because many antivirals induce errors, and the protein, the error corrector in the coronavirus will just fix it.
Yeah.
And so it makes it particularly challenging because no other RNA virus has that kind of error correction machinery.
Right. And I think that while we're on the topic of mutation, we should also just point out that people have been looking at the sequence of the virus from different patients quite a bit and making sequence comparisons to try to determine whether there seem to be some changes happening in the virus during this pandemic. And while there is some background rate of mutation, it doesn't look like there's one new type of virus that's sort of taking over and causing big changes, maybe in either the type of disease that it causes or just taking over the population. It doesn't seem like the virus is changing really dramatically. I know there have been some media reports about that, but we don't really seem to see any changes that of big substance in the population.
Yeah. And I think that's because really this virus might not need it. I mean, if you think of a virus, we anthropomorphized viruses a lot, but if you think of it just based on selective pressure, it's doing a really good job transmitting on its own. It seemingly came from the bat, very ready to infect humans. And so there just isn't, the pressure is not there necessarily to mutate and then turn into more virulence. And actually, as Vincent brings up, as viruses spread, they're more likely to develop mutations that result in less virulence or causing less disease, not more. So when you hear that in the media, viruses mutate, it's just their thing. You can just think of viruses doing what they need to do, but it's not a bad thing always. It mostly can even result in detrimental mutations to the virus itself.
Yeah, so, Brianne, you were mentioning that people are sequencing this virus from different humans. Mm-hmm. So how do we actually know that a person has a virus infection? How do we detect that?
So there are a few different ways people can do that type of thing. One of the things that we can do is we can get a sample from a person, and usually this is with a nasopharyngeal swab. You may have seen some pictures online about the swab in the nasopharynx. And then we do something called quantitative PCR, where we're able to amplify the viral nucleic acid, the viral genome, to determine whether that nucleic acid is there. And that will help us understand if the virus is present, as well as how much virus the patient has. And those are sort of the tests that are really present, that are really out there right now. There are some tests where we can look for antibodies against the virus as well, to see if someone has been previously infected. If we're looking for the virus genome, then the virus has to be there to make genome. But we might also be able to look at antibodies in the future to see if a person has been previously infected.
And can anyone speak to, there's been a lot of confusion about testing and why were we so behind with testing, and what were the hangups for why this country could not get their act together in terms of testing? Does anyone have any insights to that?
Oh boy, do we have insight.
Because it's a really big question I get about that.
First of all, we didn't have a pandemic plan. It was thrown out by the current administration, and all that would have been in it to do things like develop rapid testing. So back in January, South Korea began to develop rapid testing, and they did it really quickly, and they could see exactly who was infected, and we couldn't do that. Part of the problem is that then the CDC decided they would be in charge, and all tests had to go through them. So they initially developed a test. They distributed it. It didn't work well. And that was a big mistake, because instead of just them doing it, and I have great respect for all our colleagues at CDC, they should have said to the 50 or 100 companies in the U.S. that can do this, go develop an assay and see who comes up with one that works. All right. So that was a big problem. And then they had to make a new test. And by the time they ended up distributing it, it wasn't enough. And they finally, at some point, not too long ago, they said to the states, you can do your own diagnosis. Hospitals, diagnostic companies took it up, but it was just too late.
Right.
Those are what I can see are the reasons for this fiasco that we're in.
Right. And it's not a difficult test. I mean, each one of our labs has the capacity to do this test. If we, once you get quality control measures and once you standardize it, it's not challenging. So I think that is a really good point, that having, leadership that implements the type of movement on this and then getting it right. Now, it wasn't the primers when I say primers, these are sequences that can recognize the coronavirus. It wasn't that. Was it the enzymes that were the problem?
No, it was the primers.
It was the primers.
Yeah.
Okay.
It was the primers. One of the primers actually had a sequence that led to a really strong hairpin.
Hairpin, that's right. I remember reading. Okay.
Which means that, so when you do this process, you want the primer to bind to the virus's genome to help amplify it. And instead, the primer liked to bind to itself.
It's a problem.
Yeah. And you tell your first student, when you make that primer, make sure it doesn't fold, or else we're not going to order it. And there are many software packages that you can design primers with that'll check for that.
Week 1 of my molecular genetics class is that.
Week 1. Yep. So unfortunate, but things happen.
So we know how, do you have any more about that? Do you want to talk more about that?
Yeah, I think that's good. We should move on to
because I was going to say so we know how humans can detect infection, but how does our immune system detect the infection? Right. So now we're getting into the fun part. So what goes on with the immune response? Is that me, or do you want to talk about it, Brianne, since you're a viral immunologist?
Well, I don't know if it's would be all that different having either of us talk about it, but I can take the lead to start at least.
Okay.
So different cells in your body have some receptors that allow them to detect infection from different viruses. And so I'm right now thinking about something that's called the innate immune response, probably because, that's what I do research on. So I think about it a lot.
Me too.
And this is sort of allowing for a really early response, and a response that may be kind of broad. So these receptors might be binding to RNA from an RNA virus. They might not necessarily be super different between a flu response and a coronavirus response. Those cells will then produce some molecules, some proteins that are able to signal to other cells to change their responses or to help them prepare to combat infection. And those are called cytokines, which will influence both the innate immune cells and the adaptive immune cells.
Right. So going off of that, that's sort of like how we detected originally. And then there are a bunch of different cells, primarily dendritic cells, that will also chop up those viral fragments, and they will enable the activation of the T cells and B cells of the adaptive immune response. So it's really, I sort of think about it in 3 parts, right? So there's the innate part that sort of looks is does anybody not belong to our team? Right? Is there anybody foreign around? And then the middle part is sort of the orchestra director. So it says, okay, we've detected that here, and you need to do this, and you need to do this, and you need to do this. And so they're producing those cytokine molecules as well as presenting fragments to activate the T cells and B cells. And we've talked about how all of that happens in more detail in some of the previous immunes as well.
When these viruses, so let's say you inhale a droplet with a coronavirus and it lands on your mucosal epithelium. So what is the timing of all these things that you've talked about? How soon does it happen, and how long does it last?
Usually when we talk about sort of the initial innate immune detection, that's within maybe a couple hours after infection. I think I have a slide somewhere that says minutes to hours, but I would usually think of it as being sort of the first hour or two after infection, when that's happening. Then maybe maybe up to a couple, 2 to 4 days, will maybe start some of that process of turning on the adaptive immune system, and the adaptive immune response will then peak maybe at 7 to 10, maybe 14 days.
Yeah, which is a little different in terms of the adaptive immune system, because it takes a little bit longer. So what Brianne and Cindy had talked about is you have these reactions happening, but it's the piece of protein delivered from dendritic cells in addition to cytokines and other soluble mediators that are targeted for B and T cells. And so once the dendritic cell can present the coronavirus antigen, whatever piece it is presenting to, within germinal centers and lymph nodes. And this lymph node is really where kind of the magic happens in regards to developing an immune response and developing memory. So if you are somebody who has had an influenza infection before, you're going to have memory B cells already in your body that are ready to respond more quickly rather than something like this coronavirus where you have no memory. And so it's going to take a little bit longer to develop an immune response because this is your first time seeing this. And when we break down, there are different flavors of antibodies. So IgM, IgG, and IgA are the three ones we mostly talk about. IgE actually has some relevance to some of the abnormal immune responses that do not work well for this infection. But IgM is it's the first antibody. It responds more quickly. It tells you a little bit earlier than IgG. And it's a little bit more sticky, cross-reactive. So it kind of helps. It's polyclonal. It helps to protect somewhat, but it's minimal. Really, then once you get the dendritic cells presenting to the B cells in the lymph node, you develop these more robust IgG. And for this virus, and most coronaviruses, the ability to develop something called neutralizing antibodies is very dependent on the spike protein. There are neutralizing and non-neutralizing, and really the difference is the ability to block infection or to stop infection of the cells so that you don't have rapid replication of the virus. And what we're seeing and what we're hoping to see is the development of serological testing to see if a person has developed immunity based on IgM because it's quicker, but then we could look at IgG down the road.
And so it's interesting to note that, when I teach viral immunology, I talk about preventing infection of new cells versus eliminating infected cells.
Yes, exactly.
And
right.
so the antibodies are great at preventing new infections. And there are some things that antibodies do can help eliminate infected cells. But primarily we talk about T cells eliminating infected cells. And so those are a specific kind of T cell called a CD8 cytotoxic T cell. And so they recognize bits of the, in this case, coronavirus that might be expressed by cells that are infected, and it targets them for elimination, which we would normally think of as a very good thing because you're eliminating virally infected cells. But the problem is when you do this in the lung, or, for example, in the gut, where you have a single layer of epithelial cells that's separating you from the outside world, if you start eliminating parts of that wall, then you end up with holes in the wall. And that can be really problematic.
Right.
Mm-hmm. Yeah, and that's part of the reason in the lung where you get pneumonia, you get fluid filling the alveoli, right? And then
that's right.
Big problems.
Yeah, and it specifically then relates to this difference in mild to moderate to severe symptoms. In general, we know this virus is 80 to 82% mild, and Vincent, correct me if those numbers have changed. But the people that have more severe disease are really experiencing this immunopathology from particular cells we'll talk about in a bit, but that's what's getting people on ventilators, and that's what's destroying the capacity. I mean, basically, when you destroy the lining, as Cindy mentioned, because this coronavirus is replicating, there's no protective barrier from the edema, from the fluid of the capillaries of the lung and your air sacs. That's where the exchange happens in these cells, the air exchange. These sacs, they fill up with fluid and you're basically drowning in your own fluid. And we'll dive in a little bit more as to what this aberrant mechanism is, but it's the pathology that seems to be, and it's really happening 7 to 14 days after the onset of symptoms. And if you think of the timeline, that's really fascinating because symptoms, the incubation period for this virus is quite long. It's very different than what I know from other coronaviruses, and the fact that you can shed and not either feel sick or have very little sickness, after 5 to 6 days, then you start feeling symptoms. And then it's 7 to 14 days after that. If you're going to be severe, you're having severe lung issues. And so you think about the people who are in, on vents now, who are in the hospital having problems. They've gotten this virus 2 to 3 weeks ago. And that's a problem for healthcare. I mean, it's very hard to identify people when, and I think that's why this virus is really causing problems and why we have to be in lockdown, because it's very different than flu or other respiratory viruses.
Yeah, I don't think I really can think of other viruses that have this type of inapparent spread, and also cause this pathology.
Well, flu can be serious, right? Because it does kill up to 40, 50,000 people a year in the U.S. But I think, I'm trying to understand, in this year, the CDC estimates that we've had something like 50 million flu infections in the U.S., 700,000 hospitalizations, and about 50,000 deaths. And I know that's not the topic of this episode, but I don't understand why we can handle that and not COVID-19. And I asked a physician that, and he said, well, part of it is that we're used to dealing with seriously ill influenza patients. And also, this is all clustered. The COVID-19 patients are happening all at once, whereas, the flu is spread out over a season. I don't know if that explains it, but it's very weird.
I think it's a good point. I get asked a lot about, well, how is this worse than influenza? What's the difference? I think it speaks to, we should take influenza more seriously. We should be doing preventive measures because it does kill a lot of people. But like Vincent mentioned, it's happening all at once, and there's no immunity, and we have no stockpile of antivirals. We have no so there's no preventative or pandemic plan. It's coming all at once.
And that makes me think part of the flu issue is that we do have a vaccine and we can immunize healthcare workers to protect them, right? Whereas now we know that a lot of healthcare workers are getting infected. And, if you lose 20% of them, that's a big impact on our response.
Absolutely. Yeah, I think it is a big difference between seasonal flu and this is, as you mentioned, Stephanie, that the flu, there's a certain percentage of the population that's already immune or has some pre-existing immunity. And we can talk about that when we get to herd immunity. But this is totally novel to the human population. Not a single human had seen this before it jumped, and it's spreading like wildfire. And so it's just like those gifts that I show in my immunology class when we talk about, the effects of immunization to block the spread. It's just spreading without any barriers whatsoever. And so the only choice we have is to put the physical barriers in ourselves, which is just to separate us from the other people who might be susceptible. That's really the only thing we can do right now.
So we could, talking about the people who get the severe disease. And then we can bounce back to people who are asymptomatic, because that in itself is just fascinating, and why that's happening, I'm not really fully aware. But for the people who get severe disease, there's definitely this cycle of mediators that's causing inflammation. And as the innate immunologist, Cindy and Brianne, you can take it away. But it's a specific type of syndrome called ARDS, or acute respiratory distress syndrome.
Right. Well, I can start, and Brianne, you can fill in since I put you on the spot before.
Okay.
So I think that the main players that are happening here are that we have those neutrophils. So we talked about the neutrophil lymphocyte ratio at the beginning, the NLR, and more neutrophils is not necessarily a good thing. So neutrophils are particularly good at clearing bacterial infections, but when you put them in the lung with a viral infection, they're responding to the tissue damage that's done. They're recruited in, and they're producing a lot of inflammatory mediators and actually causing a significant portion of the damage that's done in the lung in the immunopathology. And because it's causing death of cells, those cells then lyse, they release their debris, which recruits more neutrophils and monocytes and activates macrophages, and they're going to continue that cycle of pro-inflammation. And during that process, they're producing this mucus. They're also causing the fluid leak due to the vascular changes. And then the last thing is they're producing these pro-inflammatory cytokines. And so they have systemic effects. They're going to cause the fever, but they can also cause things like blood pressure changes, things like changes in heart rate. That heart rate's also going to change due to the changes in the vasculature and the vessel leak, and the heart is trying to pump harder to keep the oxygen flowing around. There's less oxygen, so the heart and lungs have to work harder because they're not getting the oxygen. And then there's also problems with potentially clotting, and that's severe end stage things. So if the organs start to fail because of the lack of oxygen and these other inflammatory mediators having their effects, then you start to decompensate, and then the organs start to shut down, and you get into more of a sepsis or septic shock, or just a shock in general. So those are really the really severe things. The question then becomes, how do you think about manipulating those outcomes? And so people are trying to target, fortunately, because we've already developed them for other things, we have monoclonal antibody therapies that will target the various pro-inflammatory molecules. And with sepsis, certainly one of them is IL-6, or interleukin 6, is highly elevated. It's known to be highly elevated as well, and is a predictor of poor outcome in this SARS-2 infection in humans. So they're trying to repurpose existing monoclonal antibody therapies against IL-6 to try and reduce the inflammatory cascade, or what we'll often call a cytokine storm. And so that will help to interrupt that cytokine storm and reduce the inflammatory response to the immunopathology that's happening.
Yep, exactly. I couldn't have said it better in a lot of places there. Because this is, something that I think about pretty often, I've been trying to sort of keep up on a lot of the data about this. And there have been kind of 3 big papers that have come out recently, showing that those patients who are in this group who have more severe disease have more of those inflammatory cytokines that Cindy mentioned. In one case, they showed more IL-6. In one case, they showed more IL-1. And in another, they showed IL-8 and TNF. And so hopefully, or perhaps, using things like blockade of IL-6 with monoclonal antibodies could be potentially helpful, or maybe blockade of IL-1 beta, which also is one that is available. And so hopefully these studies, while some of them are pretty small, we don't have a ton of data on different patients yet. I think that as we learn more about this, we may realize that the cytokine storm is causing a lot of these problems, and maybe blocking the cytokine storm would be a useful feature.
But do we understand at all why people differ in this response? Why do some people, make this storm and others do not?
That's the holy grail of immunology.
Would you like to come to my lab?
There are so many people who would just love to know why, for example, why do some people have allergies and some people don't, right? Why do some people have autoimmune diseases and some people don't? Some of it is genetic, but a lot of it we just simply don't understand yet. And with something like this, maybe there are specific polymorphisms in genes that are important in recognizing viral infections and driving, for example, the interferon response, which we haven't talked about yet, but we probably should. But how and why certain people have more robust pro-inflammatory cytokine responses and others don't, we really don't know.
And
so I understand that there are a set of proteins that help to regulate the pro-inflammatory response. Could they be defective in some people?
Yep, yes. They could.
They could be defective, or there could be polymorphisms that just make them work differently in terms of different efficiency or different amounts of cytokines that are produced.
Is it also possible that elevated levels of viral particles could play a role in this? So some people who aren't able to control replication, or whatever reason that would contribute? Yeah,
and I think that there are a few places where that can be really difficult to tease out, and we need to be very careful when looking at some of the immune response data. Sometimes we see really high immune responses because there's a lot of virus. And sometimes we see really high immune responses because our patient differs in some aspect of their immune response.
Right. That's exactly what I'm getting at. Right. Yeah. So it's really hard to tease out. Yeah.
And what's been interesting to read some of these reports of, like Brianne said, there's been 3 recently, but they also have an increase in IL-10. Now, coming as a gut person, IL-10, we think of it more as an immunoregulatory cytokine, not necessarily one that shows up in cytokine storms. But what's been interesting about the lung pathology is not only do you have, the coronavirus is destroying a specific type of cell in the lung, then edema happens, then acute phase proteins in serum leak in, and then you have this cascade of what Brianne and Cindy were talking about of macrophages producing these pro-inflammatory cytokines. But at the same time, these macrophages are producing IL-10, which seemingly is telling the lung to lay down layers of fibroblasts. So not only are you drowning in your own fluid, but you're laying down layers of fibroblasts or fibrosis. And so now, if you don't have the edema, now you're basically suffocating. And that's been the other interesting part of this pathology for both SARS and what we're reading here is IL-10 may be contributing to the other half of lung pathology. Again, and if you have pre-existing complications with the lung, that is going to play a role in this as well. In terms of who's going to be more affected or less affected, definitely people with, lung issues.
What is interesting also is that this, ARDS and acute lung injury doesn't happen with all respiratory viruses. In fact, seasonal influenza viruses don't do that. It's only the avian H5N1 and a few others. Mm-hmm. The SARS, MERS, and SARS-2 do it, and a lot of other viruses do not. So that's another complicating factor. A person who may have a problem with SARS-CoV-2, maybe with seasonal flu would not.
Yep. Actually, in my virology class this coming Friday, we are talking about a paper that goes through that response with the 1918 flu. So, and it shows really nicely the difference between 1918 flu and seasonal flu in that response.
Yeah. Yeah.
Right. And then doubly bad is all of these things happening, macrophages and innate immune cell mediators, they are not recruiting CD4 T cells. So Cindy, right. you had mentioned, or Vincent, the ratio between neutrophils and lymphocytes. And when you have too many neutrophils, that's a predictor of severe disease and potentially death and not enough lymphocytes. And not only then not having these T cells to be able to secrete things like interferon gamma, which coronaviruses do not like, they cannot stimulate B cells. And so then you have a lesser adaptive immune response because the B cells can't secrete antibodies. So ARDS not only causes a lot of lung pathology, but then your body cannot develop the reactions that need to protect yourself even down the line.
And don't forget, remember, for every positive reaction, there's a negative reaction, right? And so one of the things that you also have with the fewer lymphocytes and fewer T cells is fewer regulatory T cells. And so they are important for regulating the level of response, right? And they're critically important in organs like the lung where, too much inflammation is really dangerous, right? So you want to control the type and the level of immune response. And so I think I've seen studies as well showing that there are reduced numbers of CD4 positive T cells going into the lung in the disease, in not in this disease because we're not looking at humans yet, but in mouse models of like SARS where they have reduced CD4 regulatory T cell influx. And so that's going to also lead to unrestricted pro-inflammatory cytokine production by the macrophages and neutrophils and other cells.
Right.
A few people have asked me whether they should exercise because apparently exercise leads to the induction of IL-6. Could someone address that?
I mean, you know what? No IL-6, right? I mean, IL-6 is still a cytokine that plays an important role in your immune system. I think what you want is a checked immune system. So if you have an increase in IL-6 after exercise, you have cells to deal with that unless you otherwise are incompetent. I think exercise is a good thing overall because when you think too about pro-inflammatory mediators, potentially weight gain, diabetes, all of those things actually can promote inflammation. So I'm not in exercise science, immunology. I don't know, maybe if anyone else has anything to think about, but
I think the only thing I would think of there is that as we've been talking about this, we've been talking about the fact that there are multiple things going on with the immune system that make SARS-CoV-2 so severe. And I think that similarly, when you exercise, you may have an increase in IL-6, but you've also got a lot of other things going on with your immune response, or with your immune system at that time, some of which may be really helpful in protecting you against SARS-CoV-2. And so I don't think we can focus just on that IL-6 with the exercise.
Yeah, that's a good point. And we know that the 3 leading comorbidities, and when I say comorbidities, meaning you're more likely to have severe disease if you have this, is hypertension, diabetes, and coronary artery disease, which a large proportion of people have. And so if you're looking at exercise as just, oh, this is bad because it's an increase in IL-6, it's really a big picture, as Brianne said. You want to try to overall stay healthy because these comorbidities are not going to help you in a coronavirus infection.
And to add to that too, I'm not sure if I know the level of exercise that's required to induce high levels of those pro-inflammatory cytokines. My suspicion is the level of exercise that any one of the 4 of us do is probably not that level. I haven't run a marathon in many years, so I can go out and run a couple miles, but I doubt that's going to do it. I think it's more intense, weightlifting or CrossFit or, marathon training that really will cause that high spike. But those people are very fit. And again, they're going to have those other regulatory mechanisms and their body's kind of used to that spike and going down. And that's sort of like a little bit of black box hand waving there. But I suspect, if you go out for your 30-minute walk 4 times a week, that's not going to cause a lot of IL-6 production. I don't think it's going to change your response to the disease.
Anything else in terms of immunopathology? We can touch on that. I mean, I know that people who have mild disease are not experiencing that. It's really people in the older age groups that are experiencing ARDS.
Right. Yeah. So I think that's the next thing we should talk about, because I think there was a lot of misinformation at the beginning, and there was an assumption that children were not affected and it was only the older people. And then we didn't understand why, because that isn't typical for diseases like this. But now we're getting numbers that are saying that, half or more of the people hospitalized are actually under 60, right? And so, but there is an age distribution to this. And Steph, you said you had some information on this? Sure.
So I think what we can say about this coronavirus is no age group is spared. And unfortunately, I don't know what these patients' underlying were they immunocompromised, but there were 3 people under the age of 18, including 1 infant, that did die within the last couple days. And so from what I know, those are the first reported cases of children or people under 18. But overall, the general trend is that a very low percentage of young people are dying. But it doesn't mean you're safe from the disease, right? You can still end up in the hospital. You could still, there is still mortality in these groups. So to say maybe that you're safe and can go to spring break if you're 22 is not correct. And actually, you're more likely to be an asymptomatic and a spreader of this virus, which particularly is a problem in children because, children gather in places like schools and daycare centers. And unlike if we're talking about, I know we don't know about stool, but if stool ends up being a way that we can transmit this virus, I mean, adult stool is well contained. Children and infant stool is, it's all over the place. So as children, as we learn more about what their symptoms are, we do know a little bit. I mean, there are some hypotheses as to why. You can kind of attack the question in 2 ways. The first way is, why are older people above 80, let's say, more likely to die? So you could attack it that way. Or why are children more protected? We know in terms of the immunology of aging that there are a lot of immune mediators that actually decrease as we age. They're particularly when you talk about T cells, we know T cells from Stanley Perlman's work, from SARS-1, CD4 T cells are really important in lung protection of this virus. And you actually have, as you age, a lower amount or less reactive amount of those type of cells. In addition to memory B cells, you're just maybe not as good as responding to pathogens as you once were. So that's called, immunoage or immunosenescence. Mm-hmm. Yeah, essentially, the effectiveness of your immune system decreases as you age.
And I think there might also be some physiologic changes in the lung that give you a little bit less lung elasticity.
Yes. And it was interesting when I was looking back at some of the previous literature, prostaglandins, which are eicosanoids, they do increase as you age, and they are also associated with increased immunopathology compared to children. That was something in animal models that different groups have shown. So just the aging of the lung in itself, and then your immune system combined with comorbidities, the trifecta of that is why we could say older people are more susceptible. But what's even more interesting about children, they have very different immune systems. And it's not like you just say, oh, children are less affected. Well, there's infants, there's children, and there's teenagers. And those are very different. Your immune system is responding very different ways. And kids, in terms of infants, they have things like maternal antibodies from their mother, other immunosuppressive proteins like adenosine. They have less acute reactive proteins in their serum, things that are supposed to keep their immune system kind of calm while the microbial environment is, the commensals are coming in the gut and in the lungs. And we don't know, but potentially that plays a role, that they have a higher ability to regulate. It's not to say that influenza doesn't cause severe pathology, but for some reason they're not experiencing the level of ARDS that adults are. There was some other thing when I was looking at the literature, other possible hypotheses, one being their children are infected with a lot of other viruses, and potentially there's competition for these viruses in the lung. I don't know, Vincent, how you feel about that. I would think that comorbidities would actually make things worse, not necessarily better.
I think it can go both ways, right? I think in some cases there might be competition depending on how similar the viruses are, right? But then, it could be worse as well. So I don't think there's one size fits all there.
Well,
right.
Could you maybe think about the fact that there also are a number of other coronaviruses that cause the common cold that tend to lead to infections in children. And so maybe there's some sort of cross-reactive immunity there.
I don't know. I would be surprised because those same infections would be in adult populations, but we don't see. We have to do the studies, but from what we know, you can be reinfected with the same common cold virus 2 years down the road. It seems to be the neutralizing antibody titers are low. You're protected for the first year or two, but then you get a more mild form of the disease. I guess that's what you're saying. I get what you're saying. Is maybe you just have more mild form and not that you don't get infected.
And maybe that you were more recently, you had one of those infections somewhat more recently.
Yeah, I could see that. That's going to be interesting to see what type of cross-reactivity some of those, because there are 4 common coronaviruses that cause colds, and it would be, it's going to be interesting to see as we move along, really with serology, what type of cross-reactivity is protective.
One of the things that I haven't really seen in the literature that I was curious about, and a couple of people have asked me about, is if you had the flu already this year, do you think you're more or less susceptible to this? And I said, I could argue either way, right? Because if you had the flu, you might have a revved up just general immune response, and maybe you can respond faster, even though it's not specific, because we know for sure that immunity to flu will not protect you from this. Those are the antibodies are specific, the Tcells are specific. There's not cross reactivity,
but maybe you'll have some extra innate immunity that could be helpful,
right? But the flip side could also be true, right? Because if you had flu and your body fought that off, maybe you have a little bit of fibrosis, a little bit of repair going on in the lung, and that might actually make you more susceptible. So I don't have a good answer for that. I could argue either way because I don't think this. The studies haven't been done. I didn't look at it. I don't know if there were studies done with SARS versus flu or anything, and see if there's different susceptibility with pre-existing infection or whatever.
The only thing that I've seen is that patients who are co-infected with both SARS-CoV-2 and influenza at the same time do rather poorly.
I would imagine so.
And so I still have told people, a flu shot's a good idea so that you don't end up getting the flu and potentially being in that co-infection group.
Right.
Oh, yeah. You don't want to go to the hospital right now if you don't have to, right?
So that's a good point. Can you actually right now get a flu shot? Will a clinic take you, or is it not?
But I think you can get them at like Walgreens and Rite Aid and those, right?
Yeah, you can. That's true. Yeah.
So I don't think you need to go to the hospital or to your doctor to get it. So it still might be a good idea.
And those are open, right? Because they're essential.
Mm-hmm. Yeah,
yeah, because they're pharmacies, right?
Yeah.
I think if it has a pharmacy, it's considered essential still. I haven't been out, so I don't know.
The other thing about kids too, we don't really know what their receptor expression is or host protease expression is. You know, ACE2 expression in adults has been defined in animal models, but not in kids. So that'll be interesting. But there's also another group of people that I have been asked about, and that's pregnant women. And you know, we hear a lot about influenza being, pregnant women are more susceptible to influenza and can actually respond quite aggressively and cause things like preterm births. And it's definitely problematic. And we don't really have the data for this virus. But what has come out is that women are not more susceptible than the general population to this coronavirus. But you don't want to end up in the hospital right now. I mean, for one, if you get if you get this coronavirus, I mean, you're giving birth to a room. I don't even think I don't know. Maybe I've heard they are not letting partners in.
Yes.
So
it depends state to state.
Okay, state to state. This is a question I get a lot. I'm actually pregnant right now. And so it's something I normally am always thinking about maternal antibodies and placentas and mammary glands, but even more so now.
Congratulations, by the way.
Oh, thank you.
Congratulations.
Congrats.
Awesome.
Yeah. It's very, very exciting. I mean, listen, if I knew this baby was going to be like gestating during a pandemic, I don't know what I would have told you. It's like a crazy time. But it is getting me thinking. You know, I remember Carolyn Coyne, who's a professor at University of Pittsburgh, said that she really started thinking about the placenta and how little we know about it during her pregnancy. So not only does my science involve this, but it's very weird to be experiencing it kind of like in real time. It's very
pandemic pregnancy.
Pandemic pregnancy. Oh. I joke with my mom and named this baby Corona. But I don't think that's a good idea. But in terms of pregnancy, I mean, there are a lot of reasons you want to avoid getting viral infections. There are reports that the cytokine storm or cytokines that are secreted, that these are soluble molecules that could potentially damage the fetus. Mm-hmm. I think that literature in terms of neuronal development is pretty, like, we need a lot more data to be able to clearly say. But for sure, in the first trimester, it's problematic. And you just don't want to end up in the hospital having to deal with this. And you can't be with the newborn. If you are yourself infected, you actually have to be separated and the baby in a NICU for that however long. So while you may not be more susceptible as a pregnant person, and the drugs that are available, they're not tested in pregnant women. So you don't really have options for drugs. And you just want to avoid it would be my advice.
I think home births are going to be a lot more common. That's a big thing here in Ithaca. You know, it's a bunch of hippies. I know a friend of mine who actually had one of those blow up whirlpool thingies delivered to their house to do a water birth at home. It's a big deal. Like,
more power to them.
Not me. I wanted to be there.
I know. I'm hoping by July that the peak has happened in May and everyone's calmed down and they have everything figured out.
I'm rooting for you. I hope that. I hope that's the case.
I'll keep you updated.
But I'm guessing that they're going to start to have separated hospitals if at all possible, right? So I don't think we have that capacity right now. But as the surge starts to wane, I would think that we would separate out if it's possible. Like here in Ithaca, it's not because it's too small. We only have one main hospital. But if you're in New York City, for example, once it starts to wane down, I'm sure you could have COVID hospitals and non-COVID hospitals.
Right. And I think that there are nurses and hospital staff who are saying outside of sort of working with COVID patients as much as possible, who are working with maternity patients and things like that.
And cancer patients. Mm-hmm. I mean, my goodness, you don't want the same nurses and doctors treating both.
No,
there is a former smallpox hospital on an island in the East River in New York.
Is it
really?
It's pretty abandoned.
Yeah, it's abandoned.
I've taken students there on a field trip 2 or 3 times, and it's really ruins of a hospital more than actually something.
At one point, for smallpox, they used to put the patients there so they wouldn't infect other people. Wow. Makes perfect sense.
But they're putting up tents and treating people in Central Park, right?
So, yeah, they even have a boat. They have a boat, so they want to offload some of the other patients to these tents and boats that don't need as much specialized care, right?
Yeah.
Right.
It just reminds me of MASH, right? The tents in Central Park.
It's very scary.
It is. Now we can talk about treatments. We have kind of a variety of topics that have been sent in to us, really revolving around questions that people have heard. Remdesivir and hydroxychloroquine are really the two main ones that I hear about. But if Vincent or if you guys want to tackle these ones.
Yeah, I'd be interested to hear what Vincent has to say, and maybe you, Brianne, too, because I think this is one of the areas where the rapidity with which things are coming out in bioarchives and things are contradicting previous studies, and studies are half done and not reviewed. It's very confusing to me to try and figure out what's real, what's not real. And I think for me, I would just want to wait until the clinical trials actually happen. But maybe you guys have some insight on that.
All right, so I'll comment briefly. Remdesivir, Mark Dennison talked quite a bit about. It was originally discovered by Gilead for Ebola virus infections, but didn't work well. It turned out in the interim to have activity against SARS and MERS coronaviruses. So it works in cell culture against SARS-CoV-2, and it's being trialed. And if you go to clinicaltrials.gov, there are a ton of clinical trials in progress. And there are two in Los Angeles in particular, this is really interesting. One is for treating severe patients, and the other is for moderately severe patients. So the key here in any therapy immediately now is to prevent people from going into the ICU, so we don't overwhelm the hospitals. So if you can treat later an infection and prevent that, that's good. So that's important to sort that out.
And it ends in vir, so it's an inhibitor, right?
So these are, remdesivir is a nucleoside analog. So it's incorporated by the polymerase, and the polymerase makes mistakes. And eventually it makes too many mistakes, and it actually chain terminates in this case. So, that hits the RNA polymerase. I think that is probably going to turn out to be useful. But as Daniel Griffin said, as long as you can treat pretty advanced patients, if you can treat someone early on, that's harder because you really want to get the ones that are coming in and they're really late in infection. Hydroxychloroquine is interesting. So remdesivir, you can use, the FDA has authorized emergency use of that. So if you have a very sick patient in a hospital, you can apply and you get approval, and it's been used many places already.
And how is the supply?
Called compassionate use, right? Oh, yep.
Yeah, I think the supply is not an issue, although it may become an issue as it's used more and more. My wife, who used to work at Merck, tells me all the raw materials to make these drugs come from China, and they are keeping them to themselves right now. So
interesting.
Hopefully that won't be an issue. So I think remdesivir, we're going to hear a lot about that in the next few weeks. Hydroxychloroquine, of course, has been in the news a lot. It's already approved for malaria and rheumatoid arthritis, and the way it works, it inhibits acidification of endosomes, so it prevents virus entry, and it's active in cells in culture against a number of viruses. So there have been some clinical trials. There's been one small one out of China, very few patients that seem to be working, but you can't really tell because they used some other treatments at the same time, so we don't know. There have been a couple of studies out of France, which have been highly criticized because they're not thought to be designed properly. But I think looking at their numbers, they are very promising. And the FDA just on Sunday, I think, said emergency use authorization of hydroxychloroquine is okay if you have a severely ill patient. And I know a number of trials are going on just this week for that. So we'll know about those two very soon, I think.
Interesting. And I think that there was a antibiotic that was paired with the hydroxychloroquine.
Azithromycin.
Yeah. Azithromycin, right. But I don't know if we really have good data on that being effective.
Yeah, I mean, that's mainly the French trial, which said the patients who had both did better. But again, I don't know.
Gotcha.
I'm not sure that you can.
I mean, you can understand why they might want to do that, right? Because viral pneumonia is predisposed to bacterial pneumonias. That's known for flu and stuff, right? So if you put the antibiotic on board, you might prevent that. But it's not clear to me, it's not my area of expertise. Maybe you guys can comment that they're ever getting to the point where they're getting to a bacterial infection. Well, it seems like this is a viral pneummonia, very acute. And there's no chance to get a secondary bacterial infection. They're just dying.
Well, what I understand is azithromycin is also an anti-inflammatory.
It is. Yeah. It is.
So maybe that's part of it. I don't know.
Interesting.
Yeah. So hydroxychloroquine is too. That's why they use it in lupus or autoimmune diseases, because it's actually an inhibitor of the nucleic acid sensing TLRs, which are inducing the pro-inflammatory cytokines. Right. So it might have some effect in that way too, not just preventing the viral infection, but actually dampening the immune mediated damage and viral recognition mechanisms and shutting down the inflammatory response. Yeah.
But just a word of warning. So one sec, Brianne, I'll let you go. I think we'll end up using some of these, but remember, resistance is going to arise, and we need to have more than 2. So there are lots more being tested, and we do need multi-drug therapy at some point.
Yeah. All I was going to say was that when I look at these data with these treatments, I think that it's promising, and it's promising to say that we need to do more studies, but I don't think that we know that we have some sort of silver bullet right now that's going to make everything better.
Right. And we talk about earlier, having a pandemic plan, we didn't have a pandemic plan. A pandemic plan would be having all of these antivirals tested for coronaviruses and stockpiled. And this, coronaviruses as a potential pandemic virus has been out there in the literature for quite some time by coronavirologists and the community. So, identifying, kind of broadly use antivirals and stockpiling them would be a pandemic plan.
Mm-hmm.
Now, in terms of treatment, we can kind of move on to our next section, which is really focusing on antibodies and vaccines and humoral immunity.
So I had one other thing I wanted to say.
And
it's sort of like that soapbox thing is I think it's critical that chemistry matters too, right? So we heard about these tragic deaths of people who didn't really know what hydroxychloroquine was, and were using things that had the name chloroquine in it, and they're using doses and purities that are not okay with human life. And it's unfortunate. And so there's a lot out there about different treatments that people are saying work or don't work. And you have to be really, really careful and just go by what the doctors say, I would say.
Yeah. Yeah, if somebody is trying to sell you something that they have the magic cure, they don't. We don't have. We don't know.
We don't
exactly,
know.
So.
That's right.
Okay, so
go ahead. So we know about the immune response, right? But so we can take advantage of this. I mean, this is the coolest part of immunology to me, right? Is once we understand how things work, we can manipulate that to help. And so we talked about one of those already, where we talked about using monoclonal antibody therapy to, for example, try to block IL-6 or other TNF or other inflammatory cytokines. But there's other ways that we can manipulate or take advantage of immune responses and how they work to use them therapeutically.
Go team immunology.
Yeah. Yay. Yeah. And so that leads to something people may have heard in the news or, within their circles. It's called convalescent serum or convalescent plasma. You may have heard it either way. And we can break that down just by the 2 different words. So convalescent it means somebody who has recovered from an illness. You're taking their serum or plasma and you are harvesting it from the blood of that person, and you're passively transferring it to another. What is contained inside that serum or plasma are antibodies. And it's a really interesting way to learn, kind of a crude way, but a way to learn about what antibodies people are generating after severe infection. And I went to look at the FDA just approved the use of convalescent serum for treatment of patients. And there's a list of criteria, one of them being you have to have recovered from the severe disease. And I think that's interesting because it's potentially suggesting that your intensity of disease or the viral replication then leads to higher titers of potentially neutralizing antibodies. So, and also the difference between plasma and serum is really just the difference between whether you use an anticoagulant or not. And if you're preparing it for passive transfer for patients, it would be plasma. And so if you've heard both, it's just because it sometimes is an interchangeable term, but it would be convalescent plasma that we'd be giving to people.
And so the basic idea here is you're taking antibodies that people have made when they've been infected, and you are giving them to someone else in order to help to get rid of or to sort of cure or reduce their current viral infection.
And it's something that's been used for a very long time. I mean, it was used in the early 1900s before we had antimicrobials. It's been used against measles and mumps and polio. So it has a history to it. But what I think more recently, people may have heard about it for Ebola, Lassa virus. It's been used, and also influenza. So there is evidence. There have been clinical trials, and some not clinical trials, but to show that this type of therapy is effective. And really in a pandemic, when you don't have time to wait around for a vaccine, this is a way to provide people with potentially protective antibodies from somebody who has recovered from the severe form of the illness.
And we call it passive immunity, right? Because it's a passive way of providing the immune response to an individual without having them develop the immune response themselves, right? As opposed to vaccines, which are an active immune mechanism because you cause the body to generate the response. So this is sort of a bypass of waiting around for the person who's infected to make their own response. You provide it for them.
Yeah, and that's sort of a little bit of the downside here. We're transferring antibodies, or we're transferring plasma that contains antibodies, and antibodies are proteins. They eventually can degrade. And we are not transferring the B cells that can keep making more antibodies. So we are kind of giving a short-term burst of antibodies, and we know those antibodies are going to degrade and not be present forever, instead of having the B cells make the antibodies themselves.
And I don't know the statistic, but when you inject the immunoglobulins, they're pretty rapidly cleared by the liver.
Yeah, they are.
And so there's very little left. So you have to inject a lot to be able to have an effect.
How are these checked to make sure they're safe and effective before they're given?
Well, so the risk of that, of course, is if you're accidentally passively transferring something you don't want people to have, which would be a virus, or something that would negatively cause effects in the person. And there are screening methods. So, people donate plasma all the time. And so they screen for blood substances that would cause an inadvertent infection with another agent, or even reactions to the serum itself. It is known that people with acute lung injury are more likely to respond poorly to passive transfer, particularly because of the blood components. And so I think that's a risk reward assessment that doctors would have to consider. But overall, it seems the literature suggests that it's safe. It's a safe practice. Oh, I thought somebody breathed.
Nope.
Well, I was just going to say, but I don't think they actually test it for neutralizing ability. We just make an assumption that if someone survived the severe form of the disease, that they have enough neutralizing antibody present in there at high enough concentration to be active.
And this is the problem with, many different infections. It's something called correlates of immunity or correlates of immune protection. We don't know how high the titers have to be to protect. SARS-1 happened so fast that we didn't do the follow-up studies maybe that we could have to try to learn that information because the virus is gone in a year or two. So I think that's something we will learn. And then there's also this, I call it a theoretical risk, because in terms of coronaviruses, it hasn't been well proven, but that's something called antibody-dependent enhancement, and it's a big topic of conversation right now. This is something really well known in dengue viral infections, is that pre-existing antibodies can make the disease worse, and the mechanism is that the antibodies are binding to certain cell types, and then make, in that case, make the virus able to enter cells at a greater ability and replicate more. So in terms of coronaviruses, I don't think it's really well founded. I think in cats, there's a coronavirus that does this, but it not so much in humans. So I think of course, it's something to think about, but it's pretty well in the coronavirus field thought that this does not happen.
Yeah, there was one paper that came out again in the past few days that was really talking about this. This has sort of led to a lot of discussion in some places. And what they're sort of arguing in that paper is that the antibodies will bind to receptors on macrophages and change what types of products the macrophages make. So it's a little different than the dengue situation. But it's really kind of one report looking at that. And they are using kind of purified antibodies there, which is different than convalescent serum, where you're getting kind of a mixture of antibodies that are coming out of a patient. And so that might also be a place where this is different as well.
And I think go ahead.
Yep, go ahead.
I think it's been criticized. Was that the rhesus paper? Was it a monkey model?
Yeah, it was a monkey model.
And I think that in terms of animal models, people are describing it as a bit of a forced infection. The viral titers have to be a certain amount, it's very high. So, and when you think about learning things in animal models, it may not always be true in humans, but it's definitely, I agree, something to think about and in terms of generating things. And it's specifically against the spike protein was what they were arguing.
Yes.
Which we know is where antibodies are generated to neutralize. So if we're targeting that spike protein because it's effective and it does cause ADE, it would be a big problem.
Right.
Yeah, I think and correct me if I'm wrong, but from some of the virologists that I've talked to, they say that this antibody-dependent enhancement is pretty easy to observe in vitro, but there's little evidence that it happens in vivo for many different viral infections, but dengue is the one seemingly exception to that where it's clear that it does happen.
Yeah, there was a lot of worry about that during the Zika outbreak, and there were a lot of observations made in cell culture saying that antibodies could enhance infection, but in people, the studies have been done. There's no evidence for antibodies enhancing Zika disease, whether against Zika virus or dengue virus, which was originally suggested. So you have to be really careful.
But one thing we do have to think about is whether when you get these antibodies, do they impact your ability to generate your own response? You were mentioning that offline, Vincent, right? So this, I think, is a question. Maybe Steph, can you comment on that? Because it seems to me that's much like maternal antibody interference, where if you already have antibodies around, does it impact your ability to generate an immune response?
Yeah, it's really interesting. And we don't know the exact answer, but the mechanism of antibody interference would be if you have antibodies that are already in and around the germinal centers, they can actually bind particularly T follicular helper cells. I know that's not a cell we've been talking about mostly, but it seems to me mechanistically involved, that they do not allow for the generation of new antibodies, and it seems to be dose dependent. So antibody dose dependent. So the more antibodies you have, the more negative interactions, and that does not promote the production of endogenous antibodies. So with infants, it's you wait to vaccinate because you need maternal antibodies to be cleared. They are protective, but the baby cannot generate its own. And so that is something in terms of convalescent plasma that has to be considered. Although the risk reward, if you have somebody who could be protected, I think that outweighs.
I mean, in a kind of simplified way, when I think about B cells making a memory response or making a secondary response, there is some evidence that naive B cells have a receptor on their surface that can bind to IgG antibodies, and that can inhibit those naive B cells. And so that can allow the memory B cells to go ahead and do their thing without the naive B cells sort of taking resources. And so there does seem to be a little bit of inhibition of naive B cells when there are antibodies present. So that might be one way that this could interfere. But I don't know, outside of the sort of classical textbook view, exactly how, what we should think about in terms of that.
But I think we're at a good point to stop this particular episode, because I know the next section is all about testing, prevention, etc.
Sure. Yeah. And wow. I didn't even realize how far we were.
I know. I just glanced over and I was like, holy wow. We've been talking for a while. But I think we still have so much more to cover. Plus, you had emails, right? I'm okay. It's up to you, Vincent, how frequently you want to try and record, but I'm okay to do it later this week. If you want to record another one or continue our conversation, it will depend on Brianne too.
Okay. Well, we'll be in touch afterwards, but this, I think today is a nice set of conversations that we don't have on TWIV for sure. So I think this will be really good to get this out.
This was awesome. Thank you guys so much for including me.
Thanks for coming. I loved our little, quad we have going here. It was a lot of fun. And I just think this episode can be dedicated to Vincent because the amount of work that you do to be able to put this all together, I'm just astounded. So thank you to Vincent. And of course, all of you for being involved.
So this is Immune number 29. You can find it on any good podcast player. And please subscribe so we know how many people are listening. If you want to see the show notes, where we have links to what we talk about, microbe.tv/immune. If you have questions or comments, please send them over. Immunology focused would be great. Immune@microbe.tv. And if you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute for how to donate $1 a month, say, and help us with our expenses. Today on Immune, we've had Cindy Leifer, who's at Cornell University. On Twitter, she is Cindy Leifer. Thanks, Cindy.
Thanks. I look forward to continuing this conversation, too. We have lots more to talk about.
We do. Steph Langle's at Duke University. On Twitter, Stephanie Langle. Thank you, Steph.
Yeah, thanks. This was great.
And thanks for organizing all of the notes and everything. I really appreciate it. From Drew University, on Twitter, she is BioProf Barker.
Yes.
Brianne Barker, thank you for coming over.
Thank you. It was great to be here. I really enjoyed this discussion.
And I'm Vincent Racaniello. You can find me at virology.blog. On Twitter, I'm PROFVRR. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back very soon.